Okay, so continuing that pattern, we have next our sarcomere. So basically what we've zoomed in a little bit from muscle fiber to now we're talking about muscle fibrils. So this is a new term here. So our scale, we went from muscle fiber, and now we're going to talk about muscle fibril, and then we can get even smaller and talk about muscle filaments. And keep this term in your back pocket, filaments, because we're going to talk about how sarcomeres contract muscle through the sliding of these filaments. So, as discussed, muscle fiber is going to be our cell. Fibrils are basically little rods made of filaments that make up our muscle cell. So this is hard to visualize. Let's see if our image up here does a good job. So again, this overall unit is our muscle fiber. Each of these rods you see here is going to be a myofibril. See that ending there? So muscle fibers are made in lots and lots of these bundles of rods, and then we can get even smaller and talk about what the myofibril is made of, and that's where we use the term filament. And so our main myofilaments, if we scroll back down, are going to be actin and myosin. These are contractile proteins that help us to contract our muscles and create joint actions and move. So they are vital to that movement component. So what does this have to do with the sarcomere? I bring it up because the sarcomere is going to be that basic contractile unit of muscle fiber. So definitely something you want to know for the lab exam. Again, the sarcomere is where we focus in on actin and myosin and how they overlap and interact to create that contraction. Histologically, we can see sarcomeres. So all of these striations you see are created by that repeating unit of sarcomere. So basically we've got one very long muscle fiber here, and then we're going to have over and over again repeating units of sarcomeres. So we're going to talk about that. We have here an electron microscope scan of a sarcomere. So we have zoomed in past the point that we are capable of with our light microscopes to see the sarcomere in a little more detail. So in terms of what I could ask you on the lab exam, I can ask you, what causes the striations that we see in this muscle fiber? The answer would be that sarcomere. But anything beyond that, anything we talk about with this electron scan is going to be not something tested, not something we can visualize with our microscopes. But let's talk about it. I know Dr. Yard does like to ask about sarcomeres in the lecture exam. So we have here different zones or bands and different lines. And so this is based on, part of this is based on how the tissue stains, and part of this is based on what the tissue we see here is made of. So to start with kind of the easiest one to identify, that'll be the sarcomere here. From Z line to Z line is one sarcomere. So the Z line is going to be that really dark stripe that you see repeating in the striations. In between, kind of the middle point of our sarcomere is going to be this guy. You can see we've got just a slight change in the staining. This is going to be our M line, the middle line. So that's literally how I remember it. M for middle. This is the middle of a sarcomere. Next up, we have some bands. So these bands are based on how light or how dark they appear on our slide. So H refers to kind of our zone where we only have myosin overlapping. So let me back up a second here. We talked about myosin and actin, and what we're seeing with the sarcomere is just different patterns of that overlap. So let's see if I can draw it up here just really quick. So basically, myosin is going to be a really thick filament. And on either side of our myosin, we'll just write an M here for myosin, we're going to have some thinner filaments called actin. And there'll be little zones of overlap, and those are the zones that we see on our sarcomere below. So basically, we've got usually two actin structures. Let me go ahead and draw on our Z line so we know the span of our sarcomere. So this is going to be a Z line and a Z line, and these are going to be our thin actin filaments. So I think I was getting ahead of myself there. So to go back down to our image, we have our M line, Z lines forming the span of that sarcomere, and then these overlaps are caused by overlaps between actin and myosin. So to go back to our H zone, this is sometimes called the heavy zone because all we have in this zone is our heavy molecule, our myosin molecule. Physically, it looks heavier than the actin because it is thicker, and that's how I remember it. We have our I bands, which are just actin. So these are going to be really light filaments, and this will make sense in a second when we connect these two ideas. And then lastly, we have our A band, and this is where we see all types of fibers. So A is going to be for all types, myosin and actin. And so what these terms actually refer to is isotropic versus anisotropic. So I know we're throwing a lot of new words in here. All isotropic means is it appears light on a slide. So if you have seen the term ISO before, maybe you're into photography or something, ISO means light. So the I band or the isotropic band is going to allow in a lot of light. And that is what we see here. It's more transparent. It's not stained as darkly on this scan. Let's contrast that now to our A band. So A actually, what it really means is anisotropic, not isotropic. So it's not going to allow in a lot of light. It is darker, and that reflects the patterns that we see here, darker stained tissue. Now, I go back to this because now that we've defined the definitions, it makes a little more sense that the I band is letting in light. It's also going to have the lighter molecule, actin only. H zone is that heavy zone. It's just myosin. And then our A band, we have all types of filaments. A for anisotropic and all types of filaments. So those are just some cues that I used to help me learn this tissue. Last part, and the reason we're spending so much time on this is because of the sliding filament theory of contraction, which is a mouthful, I know. And so we're going to take our myosin and our actin molecules and talk just briefly about how they contract to create muscle contraction. So let's redraw here. We've got a really thick myosin molecule. We're going to put it in the middle. On each side, we have a thin actin molecule. And then let's go ahead and just draw in our arbitrary Z line so we know the end of the sarcomere. And let's call this just neutral position. So this is where our muscle is not contracting, it is resting. Now we have to draw in a couple more features here. So myosin has these heads. They're called myosin heads. Let's keep it simple. And so these heads are where actin is going to bind during a muscle contraction. So from rest, they're not contracted, they're not binding to each other. But then a really cool thing that happens, if we change our color here, a really cool thing that happens is when we have calcium in our cells, calcium is going to bind to specific sites on the actin, which kind of unlocks it and allows it to create this bond with the myosin. Once this bond is created between the heads of the myosin and those sites on the actin, this rowing motion can occur. So basically, we're going to take, the myosin is going to take that actin towards the center, towards that M line, the center of the sarcomere. And the net effect, if we go back to green here, is that this entire sarcomere is going to shorten towards that M line. And this is a muscle contraction. That's the basics of it, so we're not going to spend too much time on it right now. There are more important things to know for lab, but that will come up in lecture, so make sure you're comfortable with that idea of the sliding filament theory. And then we're going to wrap it up by talking a little bit about muscle histology and some of the weird things that we can find. So most of this is not going to be testable, but again, it might come in lecture, and also I just think it's neat to know this. All of these things contribute to how our muscle system works, how it contracts, and how we can stretch our muscles and all that fun stuff. So we are going to start with the Golgi tendon organ, not to be confused with our Golgi apparatus of a generic cell. The Golgi tendon organ, and then the second term here, spindle fiber, these guys are both sensory receptors, specifically stretch receptors. And they're going to act in opposition to one another to help us monitor how our muscles are lengthening under tension, under a stretch, so to speak. So first up, we have a Golgi tendon organ. And this guy, let me tell you, this is hard to find a nice histology slide for a Golgi tendon organ. So this is definitely not something you will be tested on a lab exam. But basically, we would expect to find the Golgi tendon organ at the interface of our muscle fibers and our tendons. So these guys are found at that muscle tendinous or myotendinous junction to help monitor the stretch of muscles. So to review, muscles are attached to tendons, which insert onto bones. And so we have one of these Golgi tendons at each of those interfaces to help monitor the amount of force and the amount of stretch in our muscles. And so this is a sensory receptor, so we are talking about a nervous system structure. What you see here is an axon leading away from the tension receptor, and then not really super noticeable from a cross-sectional view, but just keep in mind this structure here, it is a nervous structure, and then let's compare that to our spindle fiber, sometimes called a muscle spindle fiber. So our muscle spindle fiber is usually found embedded within the muscle to compare it to that Golgi tendon organ. Usually they're about the size of a grain of rice, and we have about 50,000 of them in the body. So they are all over the place, helping us to regulate the amount of stretch in our muscle length. And so typically they are going to be embedded in the muscle fiber, and this is a cross section of one, so it doesn't really look like much of anything. It's just kind of an interruption to the pattern of skeletal muscle that we see here. We would expect to see more of that nervous tissue, so things like axons and dendrites would be normal to see here. And then this is a schematic of basically what is happening. So as the muscle stretches out, we have this structure, this internal structure is going to be our spindle fiber. This is going to help regulate that stretch. And so the way they work and the way these two, the GTO and the spindle fiber interact with each other is that Golgi tendon organs are going to reduce force output. They're going to reduce our ability to contract our muscles and produce force. So they are going to be an inhibitor. Whereas spindle fibers are going to enable more stretch. And so they're going to stretch to a certain point, and then we're going to be limited by the physical muscle tissue here. But the most important thing to know here is that the Golgi tendon organ and the spindle fiber are opposing each other's actions. All right, so the last nervous structure we have here is going to be our motor unit. So in this case, it's not a stretch receptor, but it still is part of our nervous system. So it is a sensory structure. Motor units are fundamental to muscle contraction. We cannot contract our muscles without having that connection to the nervous system. And so basically a motor unit is going to be a motor neuron. So we see the axon of a motor neuron here, plus all of the fibers, all of the muscle fibers that it innervates. So in this case, if we follow that motor neuron axon, we can see it has little offshoots here, let me mark them in green, going to specific muscle fibers. And so we've got motor neurons to every single muscle that we can voluntarily contract. We have different sizes of motor neurons as well in motor units. So we can have a motor neuron that's innervating just a handful of muscle fibers like we see here, or we're going to have huge motor units where we've got a motor neuron innervating hundreds of muscle fibers. So there's that wide variety that we see in the skeletal system. Okay, So again, these things might pop up in lecture, but I will not be testing you on them in lab. So it's good to know, but not need to know for lab exam 2. And then lastly, we have just a little bit of general muscle info. So we're kind of wrapping up here and stepping back, taking a step back from microscopy. So we talk about all this because it is super relevant. If you want to work in healthcare, you're going to see tons of muscle pathology, musculoskeletal pathology, or if you are just interested in working out and want to know a little more about your body, this is all good info to know. So generally we have 2 types of muscle fibers. If you take structural kinesiology or physiology, you're going to learn about some of the subtypes we have of type 2. But for now, we've got just type 1 and type 2. Type 1 muscle fibers are going to be for kind of our stamina and endurance-based activities. So they have low force output, but also low energy requirements. So they're super efficient. Type 2 muscle fibers, we can produce more force, so more strength, but we can't use them for as long. So generally, we're going to have decreased stamina with our type 2 muscle fibers. These are going to be our resistance training fibers, so to speak. And we can tell the difference between these histologically. So in the middle here, we have a mixed muscle. Now, for most of us, we're usually some combination around 50% of each type of muscle. For certain athletic sports, such as sprinting, it's really helpful to have more of our type 2 muscle fiber or high force output muscle fiber, whereas for endurance activities, like if you're trying to run a marathon, this type 1 muscle fiber is going to help you out. But for most of us, we're usually a mix of 50-50 or somewhere in there. So on the left-hand side, we have our type 1 muscle fiber. Usually this will show up as red, and you can see this, for example, you can see this in types of meat that you eat. You can look at red versus white meat, or sometimes it will show up as white meat versus brown meat. That is all related to the distribution of muscle fibers that you see in that meat. So it is common to all animals. On our right-hand side, we have our type 2. So how this works and why they're the color they are refers to things like capillary density. So our type 1 muscle fiber has to have a higher blood supply. It's oxidative, so we're going to have some substances in there to help carry oxygen to these muscle fibers. Type 2 is a little different, so we have less capillaries, and this muscle does not rely on oxygen, so we don't have as many of those structures such as myoglobin that would cause the tissue to be red. And here's a little chart. Again, not going to be asked about it on lab, but it's fair game for lecture. This talks a little more about what kind of structures or what kind of proteins we would expect to find in each type of tissue, and that is what's going to cause some of this coloration. Mainly going to be the presence of myoglobin, and then also that capillary